Hello everyone, and welcome to Chapter 8. This is the intro to metabolism. What is metabolism? Metabolism is the totality of an organism's chemical reaction. And a metabolic pathway is what begins with a specific molecule, and certain reactions happen to it until we have an end product. So we have two different types of pathways: anabolic and catabolic pathways. Anabolic pathways are what build things, and catabolic pathways are what break things down. An example of an anabolic pathway is amino acid, which turns into protein. And for a catabolic pathway, is cellular respiration, which is taking a glucose molecule, adding water to it, breaking it down into carbon dioxide, water, and ATP. So energy and the different types. So energy is the capacity to cause change, and there are different types, which are kinetic, thermal, potential, and chemical. So over here on the right, we have the girl holding a ball, which is potential energy, which is stored energy. And then we have her dropping the ball, and you see it falling, which is kinetic energy, which is energy of motion. Thermal energy over here is using heat, and chemical energy is using energy from breaking chemical bonds. So thermodynamics. So thermodynamics is the study of energy transformations, and we're going to be talking about the two laws of it. So we have the first law of thermodynamics, which is energy cannot be created or destroyed, only transferred. An example of this is the girl. As we go back to the picture, potential energy is becoming kinetic energy when she drops the ball. Energy is not being destroyed and then being put into a new form, into a new thing. Rather, it's just transferring. Thermodynamics second law. So before we go into it, let's talk about entropy. Entropy is the measure of disorder. And the second law of thermodynamics is that every energy transfer increases entropy of the universe. An example that I like thinking of is this from right here. In the beginning, it is organized, but over time, as a person using energy and moving around, going back and forth between rooms, things are being thrown everywhere, and it becomes cluttered and disrupted. So we have different types of processes. We have spontaneous and non-spontaneous processes. Spontaneous processes occur without energy input, and they can happen quickly or slowly. An example of this is a hydrocarbon being combusted with oxygen. This makes a carbon dioxide, a water, and heat. Non-spontaneous processes require energy, and they take time to happen. Free energy is a thermodynamic equation that relates entropy and enthalpy to spontaneity. Over here is the equation where delta G is spontaneity, delta H is enthalpy, delta T is measure of temperature in degrees Kelvin, and delta S is measure of entropy. So for us to have a spontaneous equation, delta G must be negative, while for us to have a non-spontaneous equation, delta G must be positive. So reactions in metabolism. So we have exergonic and endergonic reactions. Exergonic reactions are going to release heat, which makes them spontaneous, and endergonic reactions are going to absorb heat, which make them non-spontaneous. As looking at the diagram on the left, it's going to show us an exergonic reaction. It shows that we start off with the reactants at a certain energy, and then as we go up, we have this activation energy, and then once it's activated and starts going downhill, it releases energy. While in endergonic reaction, we start off with reactants at a lower energy, and then we keep adding energy to it. And then once the activation energy is reached, once it goes downhill, it does not go downhill as much. Rather, it ends up with a bigger energy. So equilibrium is a state of maximum stability. An example is this right here, where the water is flowing, but over time, it's going to go from a high level to a low level, and eventually that will balance out. In cells, that does not happen, because if that does, then a cell will die. Rather, things are always moving back and forth. So ATP. ATP is adenosine triphosphate, and it's the cell's energy shuttle. It's made by adenine, a ribose sugar, and 3 phosphates. We can hydrolyze ATP by using water to make adenosine diphosphate, which is ADP. We do this by breaking off one of the phosphate groups and adding an OH to it, which makes an inorganic phosphate. So how the cells use ATP? ATP drives endergonic reactions by phosphorylation using energy from exergonic reactions. An example of this is glutamic acid and ammonia, which makes glutamine. This reaction is non-spontaneous, and we use ATP to make it spontaneous. We do this by having the ATP molecule break and having that phosphate, which will bind to the glutamic acid, making a phosphorylated intermediate, and then we will have this ammonia go in and then make glutamine, which will then have ADP and the phosphate as a product, or as the end product from ATP. This process makes it so that the endergonic reaction becomes more exergonic, and ATP can be recycled. So when ATP can be broken apart for energy to make ADP and the phosphate, and then energy can go back into it to recycle it. So enzymes and catalysts. A catalyst is a chemical agent that speeds up a reaction without being consumed by the reaction. In this chapter, we're going to be talking about proteins as enzymes, and they are going to be catalysts. An example of this is sucrase, which is an enzyme that speeds the breaking down of sucrose into glucose and fructose. So enzymes and activation energy. So enzymes will lower the activation energy. So we will still end up with the same we will end up the same product, and our delta G will not be affected. The only difference is that the reaction will happen faster because the activation energy is lowered, which is shown by an enzyme right here. So substrate specific enzymes. So what are substrates? Well, substrates are the reactants that an enzyme acts on, and the enzyme bind to a substrate, forming an enzyme substrate complex. Over here is an example of a substrate binding to an enzyme. This is the active site, which is where a substrate will go. We see it going in here, bonding, and then we will have a product. So how can the active site lower activation energy? Well, there are 4 things. It can change the it can make sure that the orientation of the substrate is correct. It can strain substrate bonds. It can provide a favorable microenvironment. For example, like a substrate would prefer a more acidic environment, and the area around it is more basic and covalently bonding to the substrate. So what are cofactors? Cofactors are non-protein enzyme helpers. They can speed up enzymes and help speed up enzymes. So there are two types of enzyme inhibitors. We have competitive inhibitors, which bind to the active site of an enzyme, and then we have non-competitive inhibitors, which bind to another part of an enzyme, causing an enzyme to change the shape, which will not allow for the substrate to go in. This can be shown where the competitive inhibitor is going onto the active site and the substrate is not able to go in, while we have the non-competitive and it will bind somewhere else on the enzyme. That will change the active site shape where the substrate will not be able to go in. So we have allosteric inhibitors or allosteric activation. So allosteric is where a inhibitor or an activator can go. The allosteric site is where an activator and inhibitor can go, and this can change the. So for an inhibitor, it will an allosteric inhibitor will go to the allosteric site, and it will change the enzyme shape so that a substrate cannot go in, while an allosteric activator will bond to the enzyme's allosteric site, and then it will change the shape so that a substrate can go in. So we have cooperativity. So it is a form of allosteric regulation that can amplify enzyme activity. An example of this is oxygen and hemoglobin, where the more oxygens we have bonding to hemoglobin, the more and faster they will bond. So it's more of a ripple effect that will go faster. And then we have feedback inhibition, which is the end product of a metabolic pathway shutting down the pathway. So we have a reactant, and then it goes through the processes to make a product. But eventually we will accumulate a product, and part of that product will go back and become an inhibitor for the enzyme. An example of this is tryptophan, which on Thanksgiving we eat a lot of turkey, which will make us sleepy, and the reason is because of tryptophan.